in just a second, but I wanted to go through the notes real quick. I'll mark things on the notes that I would study if I were you. Let me see what color, maybe in red. I would just, for cell cycle, that's the life cycle of the cell. That's at the top. It's the life cycle of the cell. You know, all living things are made of cells. Cells are the basic unit of structure and function. Cells only come from living cells. I don't ask you a specific question about those because I'm hoping you know that. Again, the cell cycle is the life cycle of a cell. There's 3 parts to that. There's the cell division, which is mitosis. There's the growth and development phase, which is interphase. There's also that little sliver of time that's going to be cytokinesis. What are some reasons why cells divide? So why do they divide? You should know: growth, repair, development, reproduction. Remember, that's asexual reproduction. You can either have cloning, like what we would see in plants, or you could have actual splitting of cells like we see in bacteria. I gotta warn you guys, when we go back, when some people come to school in person, everybody's got to wear a mask. So I'll be wearing a mask. I know that's just like covering my face up so you don't have to see me. However, you are used to seeing me, and just please know that I'll have to wear a mask when people are here. So it'll be a little different. All right, so definitely, why do cells divide? And we're specifically talking about mitosis with this. Your genome is all of your genetic material. Chromatin is when that material is just all in strings in the nucleus. It will coil up to form chromosomes. And the reason why it coils up is to help protect the strand. It makes it easier to move the strand during mitosis. So you don't want your DNA to break, so that it's easier to manipulate it around. Make sure you know the difference between somatic cells and gametes. Somatic cells are your body cells, all of your structural cells except for eggs and sperm. And gametes are sex cells, like the eggs and sperm. The difference between the two, these have 2N for their number. These have just N. So, for example, in humans, this would be 46 to 23. It's 2 times that number. That's a big difference between the two. You only need half, so you only need N, because you're going to get N from mom and N from dad, which will give you the 2N. Okay, that's why you have fewer chromosomes in your gametes, the eggs and sperm. So definitely all these things at the top I would ask about. Remember, it's multiple choice. And if it's multiple choice and it's questions about them and there's only 10 questions total, you know, and remember, I make a bunch of questions and they're randomized. So you might get a question about chromosomes, but then not get a question about genome. You know, but these are definitely, each one is definitely included in a question I typed up. All right. What happens to prepare for cell division? The chromatid has to condense. If it's left in strings, it's easy to break. So this twisted shape protects DNA. After it gets copied, that long strand of DNA, think about like this. After it gets copied, it opens up, gets copied, closes back up. Then you're going to have it make a big pile. And then it's going to make the chromatids. These are chromatids. 2 chromatids make 1 chromosome. Okay? 2 chromatids make 1 chromosome. I don't believe a specific question about chromatids and chromosomes. Like, not what's the difference between the two. I don't think I ask anything like that. All right. Phases of the cell cycle. G1, S, and G2, these make up interphase, and interphase takes up 90% of the cell's life. That would be important to remember, that this is the longest part of a cell's life. That's something that would be important to know. 90% of the time of a cell's life. Now, there are some cells that never leave this. They, like adult muscle cells, nerve cells, red blood cells aren't going to leave that phase at all. They're going to stay in that phase until they die. 10% of a cell's life is spent in mitosis, which is actually cell division, and a teeny tiny portion is going to be in cytokinesis, which is the actual splitting. And I've got that down here in just a second in the notes. There are checkpoints. I don't ask you anything about that. Again, interphase is the longest portion of a cell's life. Mitosis is cell division. And when the cytoplasm actually splits apart, that's cytokinesis. This, something else that's important. What do you get? What's a product of mitosis? A product is you get daughter cells. You're going to get 2 daughter cells, and they have identical DNA to the parent cell. So all of them will be clones. I mean, these 2 are clones of that one, and that was a clone of another one. So really, we want all of our body cells to have the same DNA. So that would be something to remember. I don't think I asked you anything about cancer. And I'm real basic when I'm going through mitosis and what goes on in each stage. So I'm going to just highlight exactly what I asked you about each one. Okay, when does chromatin coil up? This is during prophase, because it's getting prepared to be chromosomes. Nucleus disappears, the sister chromatids form chromosomes, microtubules, they're these little puppet strings, they start to develop. I don't ask about that, I ask about the things I highlighted. Okay, the actual formation of the chromosomes. That's when the ropey chromatin coils up to make chromatids, and they get together to make chromosomes. Meta, my favorite, they line up, the chromosomes just line up. That's the important thing to remember. Meta means middle. So they're at the middle of the cell. Ana apart, think about that. This is when they actually pull the sister chromatids apart. Remember, they're identical to each other. They made copies way back when copies were made, and that's important for cell division, that all your cells are identical to each other. Telophase, this is when your nuclei begin to reform. And now cleavage is the pinching off of the cytoplasm. Cytokinesis, the cell cytoplasm actually divides. All right, so your product, you get 2 daughter cells that are identical to the parent cell where you started. Another question, okay, with plant cells, they have a tough cell wall. Remember that from when we would label? Remember, it's made out of cellulose, which is a complex carbohydrate. That's what crunches when you bite into apples and carrots. That tough cell wall here between these 2 new cells, the cell had expanded, and now you need to cut through there to build a cell plate. And so you've got to cut through cell wall here and here. You've got to build a new cell wall on each side, new cell membrane on each side. So that what's developing is called a cell plate, and it's responsible for the slicing between 2 plant cells because of the cell wall. That says tough. All right, you have a review sheet that I made for you. Oh my gosh. If you could open that up, and I'll go through the correct answers with you. Let me see how I want to do this. I'll go through the correct answers. So you can type them on there. You could number these if you wanted. You could number all of them and put them on a piece of paper if you wanted to do it that way, whatever way you want. Okay, I have to move this because it's on the crack and it's driving me nuts. Okay, that might be better. And let me turn the lights off. It gets too bright in here, and then I can't see anything because of it. All right, hope you found that. That was in the mitosis module. It's review or something, I don't know. All right, again, if you don't want to type on this, what you could do is just number each thing, and then you could write these on notebook paper. Okay, DNA, what is it? You need to know that it's genetic material. It's a code to make proteins for your cells, so they all make the same protein. Let me think something, how else? Or instructions, that's the same thing. Instructions or code to help you make proteins, because we want every cell to have the same proteins. Okay. And I'm recording this, so I will post it, and you can always review again or whatever if you missed some of the stuff. All right. What's a genome? Well, think about genome, sounds like genes. Your genome is all of the possible genetic material for an individual. So all of my DNA, whether I use it or not, whether it's cut out or whether it remains, that's my genome. And then the human genome would be the types of genetic material that all humans have. So all humans share a human genome, and we have different proteins that are turned on and off. The human genome is different from like a chimpanzee genome, which is different from a cow genome. So it's all of an organism's genetic information. That's how I worded, I believe, it could be all of a population's genetic information, all of a species' genetic information. Okay. The next one, I did have an error. You need to put an M in there. What does chromatin, why does chromatin form chromosomes? So change that to form. Remember, it makes it easier to move, and let's say maybe protection. What else could we say? It's protection. It becomes more difficult to break if you have it all coiled up. So that's why chromatin forms chromosomes. Okay. How is the DNA in a somatic cell different from the DNA in a gamete? Somatic cells are your body cells, so they're going to have a full set. They're going to have 2N. In humans, that's 46 chromosomes, where a gamete is the sex cell, the egg or the sperm, and it only has half of that number. So 2N divided by 2 is N. And that would be like comparing 46 chromosomes in our body cells to 23 chromosomes in our eggs or in our sperm. So the big thing with some of these is if you know the vocab term, then you're going to be able to come up with the answer. So if you know a soma means body, so somatic cells are body cells, then you know gametes are eggs and sperm, then that kind of answers your question for you. Okay. Okay, these are your body cells. Why would we want them to divide and make identical body cells. We'd want them to divide so your tissue could repair itself. We want them to divide for growth. So think about when you were 7, you were short, and now when you're 17, you're much taller. You grew. I don't know. I'm guessing, Will. 16, 15, 17, whatever. Development. Think about how like a little chick hatches from an egg and it develops, it changes and changes and changes and becomes a chicken. Or a tadpole is an egg that hatches, it becomes this little tadpole that swims around and it grows legs and it loses its tail and becomes a frog or a toad. That's development. So growth and development are two different things. And for asexual reproduction, if organisms want to clone themselves or we want them to clone themselves, we can take a pinch off of a plant to grow an identical plant. Or bacteria can make copies of their own DNA. They can make copies of their organelles. They can expand and split in half. And they can make new bacterial cells. So for repair, growth, development, and asexual reproduction. All right, what are somatic cells? They are your body cells. It's all your cells except for your gametes, which are eggs and sperm. We also call them structural cells. They make up the structure of an organism. So it could be in a plant, the roots, the stems, the leaves, all those cells that make those parts. In a mushroom, which is a type of fungus, it makes up the little root hairs. It makes up the body of the mushroom. So reproduction of these cells or making more of these body cells is called mitosis. And what's your final product? You start with one cell and you end up with 2 daughter cells that are identical to that parent. Okay, their DNA should be identical. Now, if there are mutations and alterations in the DNA, then you can have slight differences. That's dangerous because if you have too many differences or they're too extreme, you're not going to produce what you were supposed to produce. Okay. Longest stage of the cell cycle is interphase. That's growth and development, and the cell's just doing its thing. It's doing filtering or pumping or building things, whatever it's supposed to do. It's the longest stage of the cell cycle. Interphase. Remember, there are some cells that will, I'm not asking you specifically that, but there are some cells that never leave interphase. So once you have hit puberty, your adult muscle cells don't continue reproducing. Your nerve cells don't really after birth, they don't. And then red blood cells are regenerated every like 40 days. The adult red blood cells do not have a nucleus, so they will also remain in one phase until they die. When do chromatids pull apart? So we had and I'm going to draw this again in a minute, but in interphase, your chromosomes were like that. And in prophase, they start to form chromatids. And at one point in that cell, they pull to opposite sides. That's anaphase. Ana apart. So think ana apart. I'm not going to ask you, I mean, we used to ask like 8 million questions about these stages, and we had you label them and stuff on quizzes, and we don't do that anymore. So you lucked out. When does that chromatin coil up? It coils up in prophase. And I can't think of a way to remember that. Coils up in prophase. What is the cell plate? It's going to be in plant cells. Pro move and coil up. Pro. You're producing your chromosomes. All right, cell plate is going to be a tough organelle that develops because it cuts through the cell wall and the cell membrane and forms new cell wall, new cell membrane when you're dividing plant cells. And that's because of that cell wall in plant cells, because it's so tough. It's made of tough carbohydrates, and it has to be cut. When do cell nuclei reform? So at the beginning of mitosis, the nucleus dissolves, and it has to dissolve so those chromosomes can move. Well, at the end of mitosis, the nucleus or nuclei can reform, and that's going to be in telophase. Yes, I will put all of it in Canvas, Sam, and the recording will be in Canvas. Sure, no problem. All right, down at the bottom, and you know what? Look at my messy mess. So I'm going to take you write it down there or on a piece of paper. I'm going to use a new piece of paper because that was a hot mess down there. All right. Okay. La, la, la. 90% of the time, the cell's just doing its thing. That's interphase. Let's say this is a cell, maybe you scraped your knee and you need to make new skin cells. So something's going to tell the cell that it needs to go through mitosis. And remember, that's that teeny tiny, okay, this is 15, 20, or I'm sorry, 15, 30, to be about that much. Mitosis. So something tells this cell, hey, you need to make more tissue. So what's going to happen? The cell nucleus is going to start to dissolve, and that scrambled up pile of chromatin is now going to coil up to make chromosomes. This is prophase. Meta, middle. And they can fill up in either way they want. Remember, they're copies of each other, so they can line up in either way. This is Anna apart. All right, so they start to come unraveled. Whoops, here. There's this pinching off of the cytoplasm. It's the nuclei begin to reform. The chromosomes, or chromatids, I'm sorry, start to unravel to make the chromatin again, so it looks like spaghetti. This is telophase. And finally, we end up with, what's it look like up there? The actual splitting is cytokinesis. All right, I am not having you label this. Instead, I would ask you specific questions. So the questions could be, when do the chromosomes line up at the center of the, or at the equator of the cell? When do the sister chromatids separate? When are they pulled apart? When does When does the chromatin coil up to form chromatids? When do the nuclei start to reform? When is the cytoplasm split between the two daughter cells? All right. Are there any questions? So this review sheet is really, really, really going to be helpful. It's the kind of thing that when you take a quiz, if you have two devices open, you can have one on one, one on the other. Top of the back page, the drawing, and I'll scan that in too. Now, a hint, you're taking the quiz during class on Friday. Okay, I'll spend the first 5 minutes or so if you want to review or ask me a question, and then I'm going to give you about 15 minutes and let you take it during class because it should be fairly easy for you. You have everything you need, and I just went over every single question. Okay. If you don't have any questions, we're going to continue and now go into cell differentiation. So we've got the cells all divided, but now what? Like, how do we go from, one cell to a liver and a heart and a bladder? Like, how does that happen? So we gotta talk about that. Let me get some white blank paper. I'm trying to clean my desk off for next week when kids come back, and then once I clean it off, I can't find anything. Oh, I knocked stuff over. All right, I've got blank paper because I want to kind of go through and remind you what's going on here, okay? So I will scan that in with wherever my note pages are, and that review. Okay. It'll be this afternoon before I get a chance to do that, but I'll scan it in for you. Okay. I just wanted to kind of backtrack so we all are thinking the same thing here. Do you remember the fact that there is one egg? We haven't talked about how it got made, but there's an egg from the ovaries of a female. There's a very tiny sperm from the male. Remember, the egg, if it's human, that's going to be 23 chromosomes, and if it's human, it's going to be 23 for the sperm. And those two get together. Thousands and thousands of sperm make it all the way to the egg, but only one will break through. And when that one breaks through, its DNA fuses with the DNA of the egg. Okay, that's just the way it works. It's a rule. Only one of them gets there. And when it does, then there's 46. So you have 46 chromosomes. When it's going through this, within a couple days, it depends, in a human, it's different from other organisms. But that one cell is going to go through mitosis and make an identical cell. Through mitosis, okay, and they're all the same. This is going to continue. You're going to have 4. We're going to split into 8, 8 into 16, 16 into 32. This is going to continue and continue. And as this is developing, at some stage in the development of that embryo, there is going to be a hollow ball. Okay, what's going to happen is it's going to look like a ball of cells, but it's actually a hollow ball. When it's back up here, like that, that's called, I'm trying to find a marker to mark this for you. When it's this ball, it's called a morula. When it's this hollow ball, it's a blastula. So we'll say the embryo is at the blastula stage. The embryo is at the morula stage. In person, if you were here, we could actually look at these things under the microscope. We could look at different fish embryos, and we could see these hollow balls under the microscope, on cells under the microscope. So we have this hollow ball of cells, and the best way I've ever seen this described is when I was student teaching, I saw a teacher actually take like an old volleyball, and it must have been pretty weak, because he had it and showed this hollow ball, like the volleyball is hollow on the inside. That's the blastula stage, and he took his fist and he punched it. That's what happens to this hollow ball. As you're developing as an embryo, that hollow ball is going to cave in. So pretend that looks like a caved in volleyball. This is called the gastrula stage. Okay, that's the gastrula stage. And there's going to be some other This is going to continue till it looks more like kind of more like this. Oh, you're blinded right there. That's going to be that early embryo. And there's going to be 3 layers here. They're going to make the 3 parts of that developing embryo. All right, when we're looking at this, the tissue on the outside of the gastrula, this is going to become the ectoderm. Ecto means outside, derm, layer. That's what that means. The inside layer in here, inside that, where it's crushed in or pushed in, that's going to be the endoderm, the inside. And this in here, this hollow area, mesoderm.
She better post that because she doesn't. Lily, I'm gonna become someone with you.
What?
Why do we have so many quizzes? Like, that sounds so.
You guys got some problem? Somebody got a problem or a question? Okay. I hope you weren't complaining about me. So these are going to become your 3 layers. You are going to have a project, okay? This is the same one the other honors bio is having. You are going to have a project showing this, going from one egg, one sperm going through mitosis to this. Okay. I will walk you all through that. Do you think I walked you through that last CER? That's okay, Sam. Did you guys feel like I told you like how to prepare for the CER? I haven't graded all of them yet, so I haven't posted the scores. So I promise you, I'm going to walk you through how to do this project. And then the project will also be a summative score. So it should be easy for you to get an A on it. Okay, so these are going to become the 3 layers. In the notes today, I'm going to show you what they become. Meso, think about our insides of our bodies. Meso, middle. That's going to be some organs on the inside. Okay. Ectoderm, skin and hair and nails. So these different layers are going to become different parts. And all of this is called differentiation. So it's how those cells can differentiate. Even though they all have the same DNA, only parts of their DNA get expressed or read. So only parts of their DNA tells them what kind of thing to make. Oh, I got a hair. Okay. And I will post this for you, but it probably won't be till this afternoon. We do have a job interview. We have to interview for a new teacher from like 12:30 to 1, I think. So it'll probably be after that because like 11:30, I usually run home. It'll be like after 1 o'clock before I can post this extra stuff. Okay. All right. For cell differentiation, I did give you guys some notes. If you, wait, ball of squish, where at? There? Where? Right there? Morula. I just think they're cool names because it sounds like the names of, I don't know, like if somebody wanted to be a witch for Halloween, you know, you're Morula the witch or something, or Blastula. Sounds like something from Harry Potter. Okay, Pokemon. I don't know any Pokemon names, so I can't make that reference. Guys, got all this before I start the notes. All right, you can do one of 2 things, or well, actually, there's a couple things. You could type on the blank one. Just a second, Sam. You could type on the blank one. You could write this on notebook paper, or you could follow along and annotate and then go back. Okay, so these are the stages. So this is called, when it's just a ball of cells, it's called the morula stage. So somebody might, they might be talking about, this mutation occurs at the morula stage of the embryo. Then you'd know that something happens here to alter the DNA. Okay, this is called the blastula stage, that empty ball. So you could say during the blastula stage, the egg makes its way down the fallopian tubes to the uterus. The gastrula stage is when it really starts to form what we're going to see as the future embryo, because I'm going to show you. Look at how it's shaped like this. An embryo is kind of shaped like that too. Okay, this would be an embryo, and see, it's got like a spine along the back. Vertebrate animals have almost identical embryos, and that's something when we study evolution, it's going to be really tough for us to see the difference between like a sheep, horse, whale, human, bird. All the embryos look very, very similar because see this shape? That's what's going to become this embryo. And that embryo will further change to have like little limb buds. It'll keep changing and changing. It does have little things here. We all have gill slits when we're embryos. Isn't that cool? You found pictures of yourself as a fetus. That's awesome. We didn't do that when I was a kid. I can't imagine how cool that is to have a picture of yourself before you were even born. That would be really cool. All right, now do you guys have all the names here? Sam, did that make sense that those are the stages? So it would be similar to saying this is preschool, kindergarten, high school, something like that. Okay? All right. So in your notes, it should look like this. Cell differentiation. Let me see, just a second. And cell differentiation means that all of your organism's body cells have the same DNA, but they don't make the same proteins. We all begin as one fertilized egg that is we are produced through sexual reproduction, where one egg and one sperm come together. But now think about that. We are one cell. What happens to that one cell? So then we have 200 different kinds of cells in our bodies. So we go from one type to 200 types. So we're one type, and we go all the way to have it being made of 200 types. I think that's pretty amazing. And that can only happen through this cell differentiation, where certain genes are turned on and off. Okay, we're born with all of this DNA, but we only use parts of the DNA to make different things. We don't use that whole long strand to make everything. In fact, we've got about half of our strand that we don't think we use at all to make things. We actually use it as like on-off switches and stuff. How do all of the different cells and tissues form? Now remember, this is going to be online, and these are already scanned in. How do all of the different cells and tissues form? And like I said, we've got over 200, or it's around 200 types of cells. We've got about 12 or 13 body systems, depending on how we count them. And each body system is made out of a bunch of organs. And those organs are made out of tissues that are just cells all doing their jobs. So again, I'm showing you, just like we drew a second ago, or I drew to show you, that we get that one fertilized egg that has a complete set of chromosomes, and then it splits into 2 and splits into 4, it splits into 8, and it continues and continues and continues. What time do we get out? 10:25. All right, we got 10 minutes or so. Okay, hope you got all that. Remember, I'm going to put it online anyway. Okay, in those early stages, but this differentiation can happen at any time during life, which that's going to lead to stem cells, where some of our cells can become any kind of cell. We'll talk about that later. But in these early stages, the cells could become any type of body cell. It's just whether they're triggered to read certain DNA or to leave that turned off. So this, with stem cells, it could happen at any time in our life. We are typically not going to have our liver start developing an ear, when we're growing, when we're sitting around here. Okay, so typically we're just going to keep our cells at whatever they're doing. There could, I guess, be a mutation where it would start growing the wrong tissue. So in those early stages, they can become anything. There's nothing saying that these, like in that ball of cells, there's nothing saying, oh, this one has to be an ear, and this one has to be a knee, and this one has to be blood. There's nothing that says that in those early stages. Those are called, when we're in those very early stages of an embryo, those are embryonic stem cells. Will we probably only contain about, I'm just guessing, around 70% of the same DNA as a penguin? Because I think it's 80% as a cow. 80% we have in common with a cow. So what's more likely could be something similar to like a monkey or a chimp, because we have more in common. And so really part of our DNA could be turned off, or part of their DNA could be turned on or off, and we could become more like them. We could maybe have more hair covering our bodies or something. But we're not truly, I don't think we would ever produce a different species. I think it would die. I think that it just would never develop. I mean, I guess that's possible, but I don't know. These early cells are unspecialized. They don't have a job yet. You know what? While you're writing it down, I'm going to look up, because you guys always ask me questions that are pretty cool. So just a second. What organism shares the least DNA with humans? Maybe a fungus or something. Let me see. What ones are we least? Oh, sponges and comb jellies. That is very cool. Oh, well, even protists like amoeba and paramecium. We share 50% of our, oh my gosh. We share 50% of our DNA with a banana, 85% with a mouse, 61% with a fruit fly. That's very cool. Very, very cool. So these early cells are unspecialized cells. So they could become anything in that early embryo. Cells or make new ones. And they can develop into any of the specialized types of cells. So they could become body cells. They could just become, you could keep producing more and more and more, and they can make like any of those same cells until you get a larger mass. Or they could begin specializing. I want to look that up and see at what point do they. All right. It looks like sometime after the 5th day. During the 5th day of development, which is amazing,